Hi, welcome to my YouTube channel. This is Soumya. In this video, I'm going to solve a KSET license question paper from 2014 and this is the part 4th video and I'm solving the paper 3. So before that, I would like to ask, how was your exam? Just 2 weeks back you have given the exam. I'm so excited to know how you all done the exam. So comment in the comment box so that I will get to know. And many people have requested to solve the question paper. Sure, I will do it, but I didn't have a question paper, so it got a bit delayed. And I will make sure I will upload the solved question paper. Now just don't think about result. ಅದ್ರ ಬಗ್ಗೆ worry ಮಾಡ್ಕೊಂಡು ಕೂಡಬೇಡಿ. And yeah, so ನೀವು prepare ಚೆನ್ನಾಗಿದ್ರೆ exam ಚೆನ್ನಾಗಿ ಬರ್ದಿರ್ತೀರಾ. So ಸ್ವಲ್ಪ doubt ಇದ್ರೆ no, don't worry. So you can give the next attempt and don't fear of failure. And because failure is the one that make you strong and that make you to realize where you step down. In failure inda you will get to know so ಏನೆಲ್ಲ ಮಿಸ್ಟೇಕ್ ಆಗಿದೆ, ಸೊ ನಾನು ಏನು ಕರೆಕ್ಟ್ ಮಾಡ್ಕೋಬೇಕು ಅಂತಂದೇಳಿ ಫೇಲ್ಯೂರ್ ಇಂದನೇ ಗೊತ್ತಾಗೋದು. You are going to learn from failure. Okay, failure is the one that make you to correct your mistakes and move towards the success. So without failure you are not able to learn anything. Okay, keep that in mind and don't stop preparations. Preparation is very important because if you are in this field means unless until you are going to pass it, don't drop it. Okay, so that I would like to say. Now let's begin with this video. 33 question. Cancerous cells are destroyed by following type of cell. Option A, macrophages. Option B, NK cells. Option C, neutrophils. Option D, eosinophils. Cancer cells ಯಾವ type of cells ಇಂದ destroy ಆಗುತ್ತೆ ಅಂತ ಕೇಳ್ತಾ ಇರೋದು. And you know all these 4 options are nothing but these are types of WBC only. And first one, macrophages. What are these macrophages? Macrophages are a type of WBCs, and as in the name indicates, macro means large or a big. Okay, phase means eat, and these are professional phagocytes. Do you know what is phagocytes? These are going to eliminate the foreign substances by engulfing the foreign materials. So macrophages functions like engulfing and digestion of the microorganisms, and these are highly specialized in the removal of dying or dead cells, or you can say cellular debris. So these macrophages have nothing to do with the cancer cells. So hagage option A is not the correct answer. Option B, NK cells. What are NK cells? Again, these are also type of WBCs only. NK means natural killer cells. Okay, natural killers are WBCs that destroy infected and diseased cells, infected and diseased cells like cancer cells. Okay, these are also called as effector lymphocytes. Okay, hagagi maybe you might get the answer now. So option B is the correct answer. In natural killer cells are the ones they have the capacity to kill the cancer cells. Okay, destroy the cancer cells. And coming to the option C, neutrophils. Neutrophils, these are the type of WBCs that help the body to respond to infection and damaged tissues. Okay, that's the function of the neutrophils. And option D, eosinophils. Eosinophils, these are also type of WBCs that protect your body from parasites, allergies, and foreign bacteria and outside organisms. Okay, that is the function of the eosinophil. For answer for the 33 question is the option B, that is natural killer cells. Okay, I hope you understood. Now let's move on to the 34th question. Phagocytes may be characterized by which of the following statements? Option A, it involves fluid uptake by small vesicles. Option B, it involves uptake of cellular debris in large endocystic vessels. Option C, it is not important in digestive process in mammals. Option D, it is constitutive process. First we will know what are phagocytosis. What is phagocytosis? Phagocytosis is the process by which a cell uses its plasma membrane to engulf a large particles and giving rise to an internal compartment called phagosome, and it is going to digest it. It is going to collect the foreign materials or debris or microorganisms and it is going to ingest it. ಹೇಗೆ ingest ಮಾಡುತ್ತೆ? It gives a internal compartment that is called as phagosome. In the phagosome, those are foreign particles, microorganisms are all destroyed. Phago means engulfing a large particle ಅಂತ ಹೇಳ್ಬೋದು. Okay, so ಇವಾಗ ಆಪ್ಷನ್ಸ್ ನೋಡೋಣ. Involves fluid uptake by small vesicles? No, option A is not the correct answer. It involves the uptake of cellular debris in large endocystic vesicle. ಅಲ್ವಾ? So definition ಕೂಡ ನಾನು ಅದನ್ನೇ ಹೇಳಿದೆ. So ಹಾಗಾಗಿ option B is the correct answer. It involves the uptake of cellular debris in large endocystic vesicles. Okay, option B is the correct answer. Now we will move on to the 35th question. Consider the following statements. IS elements are option A cut and paste transposons, option 2 these are sequence of DNA in genome of prokaryotes, option 3 these sequences are parasites of host genome. Which of the above statements are correct? Option A 1 and 2 are correct, option B 3 and 1 are correct, option C 2 and 3 are correct, option D 1, 2, 3 are correct. First we will know what are IS elements. Do you have any idea what are IS elements? Full form ಹೇಳೋದಾದ್ರೆ IS elements means insertion sequence elements. Okay, these are insertion sequence elements. The insertion sequence element is a short DNA sequence that acts as a simple transposable elements. Now the question is, what are transposable elements or what are transposons? Okay, transposons are also called as transposable elements, and it having the another name. It is also called as jumping genes. What are these jumping genes or transposons or transposable elements? As in the name indicates, transposons. Trans means movement, transport anta heltivalla, moving from one place to the another place. So the same thing these transposons are doing here, but not a different location. Okay, here what they will do, they are going to jumping or moving from one location on the genome to another, and how they are moving, that can be possible by cut and paste. And these transposons, they carry only the genes coding for transposase enzymes. And also in simple, if you have to remember, you can just call them as mobile genetic elements. Mobile means movement. Okay, they are moving from one genome to the another genome. Okay, first ಏನಾಯ್ತು, ಫಸ್ಟ್ ಆಪ್ಷನ್ ಕಟ್ ಅಂಡ್ ಪೇಸ್ಟ್ ಟ್ರಾನ್ಸ್ಪೋಸನ್ಸ್ ಅಂತ ಹೇಳಿದೆ. ಓಕೆ, ಸೊ ಮೂವ್ಮೆಂಟ್ ಆಗಬೇಕು ಅಂದ್ರೆ ದೇ ಶುಡ್ ಕಟ್ ಅಂಡ್ ದೇ ಹ್ಯಾವ್ ಗೋನ್ ಟು ಪೇಸ್ಟ್ ಮೀನ್ಸ್ ದೇ ಹ್ಯಾವ್ ಟು ಗೋ ಅಂಡ್ ಜಾಯಿನ್ ವಿತ್ somewhere else. Okay, so and second, these are sequence of DNA in genome of prokaryotes. Okay, so ನಾನು ಹೇಳಿದೆ these are the small segments or short DNA sequence ಅಂತ ಹೇಳಿದೆ. Okay, so that is also correct. In the genome of prokaryotes, even you can find these IS elements in the eukaryotes as well. So option 2 is also correct answer. And coming to the option C, third one, these sequence are parasites of host genome. What are parasites? Parasites means parasites are those organisms which depends upon the another organism for the food and shelter, right? So here ಏನು ಹೇಳ್ತಾ ಇದ್ದಾರೆ, ಈ IS elements are parasites of the host genome ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. Yes, these are parasites of the host genome because they uses the host machinery for their own amplification. ಅಂದ್ರೆ ತನ್ನ ಒಂದು ಸೀಕ್ವೆನ್ಸ್ ನ ಡಬಲ್ ಮಾಡ್ಕೊಳಕ್ಕೆ it is using the host machinery for their amplification. ಹಾಗಾಗಿ option 3 is also a correct answer. So now 3 ಇರೋದು ಯಾವ್ದ್ರಲ್ಲಿದೆ? That answer there in the option D. So option D is the correct answer. Correct answer ಬಂದ್ಬಿಟ್ಟು option D. Now let's move on to the next question. 36th question. Which of the following is not involved in DNA damage repair? Option A excision repair, option B recombinational repair, option C SOS repair, option D RNA polymerase. Which of the following is not involved ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಇಲ್ಲಿ ಈವನ್ ನಿಮ್ಗೆ ಈಸಿಯಾಗಿ ಒಬ್ರೊಬ್ರು ಡೆಪ್ತ್ ಆಗಿ ಓದ್ಕೊಂಡಿಲ್ಲ ಅಂದ್ರೆ ನೀವು ಈಸಿಯಾಗಿ ಇದನ್ನ ಕ್ವಶನ್ ನ ಈವನ್ ಸಾಲ್ವ್ ಮಾಡಬಹುದು. ಇಟ್ಸ್ ಫಾರ್ ದ ಪೀಪಲ್ಸ್ ಹೂ ಡಿಡೆಂಟ್ ಸ್ಟಡಿಡ್. ಅಂದ್ರೆ ಸುಮ್ಮನೆ ಜಸ್ಟ್ ನಿಮ್ ನಾಲೆಜ್ ನ ನಿಮ್ ಸ್ಮಾರ್ಟ್ನೆಸ್ ನ ಯೂಸ್ ಮಾಡಿ ಸಾಲ್ವ್ ಮಾಡಬೇಕು ಅಂತ ಆನ್ಸರ್ ಮಾಡೋರಿಗೆ ಇದನ್ನ ನಾನು ಹೇಳ್ತಾ ಇರೋದು. ಬಟ್ ಆಕ್ಚುಯಲ್ ಮೆಥಡ್ ನ ಹೇಳ್ತೀನಿ. ಸೊ ಇಲ್ಲಿ ನಾಟ್ ಇನ್ವರ್ಡ್ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಇಲ್ಲಿ ಎಲ್ಲಾದ್ರಲ್ಲೂ ರಿಪೇರ್ ಅಂತಾನೆ ಇದೆ. ಹಾಗಾಗಿ ಅಂಡ್ ಆಲ್ಸೋ ಒನ್ ಮೋರ್ ಥಿಂಗ್ ಯು ಹ್ಯಾವ್ ಟು ಅಬ್ಸರ್ವ್ ಈಸ್ ASE. ಓಕೆ, ASE ಮೀನ್ಸ್ ಎಂಜೈಮ್. ಹಾಗಾಗಿ ನೀವು ಬ್ಲೈಂಡ್ ಆಗಿ ಹೇಳ್ಬೋದು ಆಪ್ಷನ್ ಡಿ ಇಸ್ ದ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಬಟ್ ಆಕ್ಚುಯಲ್ ಸೊಲ್ಯೂಷನ್ ಇವಾಗ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡ್ತೀನಿ. ಫಸ್ಟ್ ಆಪ್ಷನ್ ಎ excision repair. What is excision repair? Excision repair ಅಂತಂದ್ರೆ it corrects the small DNA lesions, and there are different types of excision repair. Those are all called as 3 different types are there. Those are called pair excision repair, nucleotide excision repair, and mismatch repair. Okay, these are the main 3 types of excision repair. What is this excision repair? It is going to remove the part of the damaged DNA strand and replace it through a DNA synthesis using the undamaged strand as a template. So ಹಾಗಾಗಿ option A is the correct answer. Okay, and coming to the option B, recombinational repair. As in the name indicate, recombinational means ಮತ್ತೆ recombined ಆಗುತ್ತೆ ಅಲ್ವಾ, ಅದೇ ಅಲ್ವಾ ಮೀನಿಂಗು. ಇಲ್ಲೇನಾಗುತ್ತೆ, is a mechanism for generating a functional DNA molecule from a 2 damaged molecules. ಸೊ 2 damaged molecules ಇರುತ್ತೆ. ಆ 2 ಡ್ಯಾಮೇಜ್ಡ್ ಆಗಿರ್ತಕ್ಕಂಥ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಲ್ಲಿ ಅವೆರಡನ್ನು ಕಂಬೈನ್ ಮಾಡ್ಬಿಟ್ಟು, ಸೊ ರೀಕಂಬೈನ್ಡ್, ದಟ್ಸ್ ವಾಟ್ ದ ಮೀನಿಂಗ್ ಹಿಯರ್. ಕಂಬೈನ್ ಮಾಡಿಬಿಟ್ಟು ಫಂಕ್ಷನಲ್ DNA ಪ್ರಿಪೇರ್ ಆಗುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ಆಪ್ಷನ್ ಬಿ ಇಸ್ ಆಲ್ಸೋ ಎ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಕಮಿಂಗ್ ಟು ದ ಆಪ್ಷನ್ ಸಿ, SOS full form here is that save our soul. This is also a DNA damage repair mechanism, and it uses a important protein name called as REC-A protein. With the help of that, this DNA damage repair mechanism is going to start. Option C is also a correct answer. So which is the one which is left? Option D, and it is an enzyme. So hagagi option D is the correct answer. Moving on to the next question, 37th question. Multigene family evolved through option A only gene duplication, option B random mutation, option C only unequal crossing over, option B both duplication and unequal crossing. First we will know what are multigene families. Multigene families are nothing but these are multigene families are a group of genes that have descended from a common ancestral genes. Therefore they have the similar functions and similar DNA sequences. So they have the similar DNA sequences and similar functions. Why? Because ಅವು ಏನು ಕಾಮನ್ ancestor genes ಆಗಿರ್ತವೆ ಹಾಗಾಗಿ. ಸೊ ದೀಸ್ multigene families evolve through what ಅಂತ ಕೇಳ್ತಾ ಇದ್ದಾರೆ. Obviously if they have to evolve, option D is the correct answer. They both duplication and unequal crossing over adre matra they can evolve. Moving on to the next question, 38th one. Antibiotic resistance among bacteria represents option A balancing selection, option B stabilizing selection, option C directional selection, option D disruptive selection. So first we will understand what is antibiotic resistance. Antibiotic resistance andre easy agide. Antibiotic resistance ಅಂತಂದ್ರೆ ನಿಮ್ಮ ಬಾಡಿನಲ್ಲಿ ಏನೋ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದೆ ಆಯ್ತಾ, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಿಲ್ ಮಾಡಕ್ಕೆ ಡಾಕ್ಟರ್ ನಿಮ್ಗೆ ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ಕೊಡ್ತಾರೆ. ಬಟ್ ಅದು ರಿಪೀಟೆಡ್ ಆಗಿ ನೀವು ಇಯರ್ ಬೈ ಇಯರ್ ಇನ್ಫೆಕ್ಟ್ ಆಗ್ತಾನೆ ಇರ್ತೀರಾ, ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ತಗೊಂತಾನೆ ಇರ್ತೀರಾ. ಅವಾಗ ಏನಾಗುತ್ತೆ, ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನು ಇಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಗೇನ್ ಎ ಸ್ಟ್ರೆಂತ್. ಸೊ ಆಂಟಿಬಯೋಟಿಕ್ ಹಾಕಿದ್ರು ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಿಲ್ ಆಗಲ್ಲ, ಅಂದ್ರೆ ಇಟ್ ಬಿಕಮ್ ರೆಸಿಸ್ಟೆಂಟ್ ಟು ಇಟ್. ಅಂದ್ರೆ ಆ ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ಏನು ಅದ್ರ ಫಂಕ್ಷನ್ ಏನು ಟು ಕಿಲ್ ದಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಬಟ್ ಅದೇನ್ ಮಾಡುತ್ತೆ, ಆ ಕಿಲ್ ಮಾಡೋದನ್ನ ಓವರ್ಕಮ್ ಮಾಡ್ಬಿಡುತ್ತೆ. ಸೊ ಅದನ್ನ ಹಾಕಿದ್ರು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಾಗ್ಬಿಡುತ್ತೆ, ಇನ್ನೂ ಸರ್ವೈವ್ ಆಗುತ್ತೆ. ಓಕೆ, ಅದನ್ನ ನಾವು ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅಂದ್ರೆ ಇಟ್ ಗಾಟ್ ಎ ಪವರ್ ಟು ರೆಸಿಸ್ಟ್ ದಟ್ ಆಂಟಿಬಯೋಟಿಕ್. ಇದು ಯಾವುದಕ್ಕೆ ಇದು ಏನನ್ನ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡುತ್ತೆ, ದಟ್ ಟು ಅಮಾಂಗ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಓಕೆ, ಟು ಆನ್ಸರ್ ದಿಸ್, see here, DNA mutations happens randomly during division by mitosis. So we know this, right? So some of these mutations code for antibiotic resistance. In case of bacteria, I'm talking about the bacteria here, okay? So the DNA mutations happens randomly during division by mitosis, means during cell division, during mitosis, mutations ಆಗುತ್ತೆ. Some of these mutations are going to code for the antibiotic resistance. Okay, when a bacteria colony is exposed to antibiotic, most bacteria without the resistance is exposed to antibiotic. Most bacteria without the resistance, mutations will die. Okay, and the antibiotics are a selection pressure here. Okay, as a result, the chances of surviving and dividing to create an offspring over many generations, antibiotic resistant mutations will become more frequent. This is a directional selection. So because antibiotic resistant mutation will become more frequent, so idana navena helbodu, this actually represents a directional selection. Okay, that actually represents a directional selection. So option C is the correct answer. Moving on to the next question. MHC class 1 molecules represents peptides to T cells in which pathway? Option A endocytic, option B humoral, option C complement, option D endogenous. Immunology ಬಗ್ಗೆ ತುಂಬಾ videos ಮಾಡಿದೀನಿ. If you are interested, please do watch those videos. I will definitely say that those videos are going to help you. And here MSC class 1 cells, actually they are going to represent the peptide T cells in an endogenous pathway. Okay, so endogenous pathway nalli represent ಮಾಡುತ್ತೆ. Okay, that is the option D is the correct answer. Now we will move on to the next question here. 40th question. What is the meaning of molecular clock? Option A, rate of DNA or protein sequence evolution is constant over time or among evolutionary lineages. Option B, rate of only protein sequence evolve in constant over time or among evolutionary lineages. Option C, rate of only DNA sequences evolution is constant over time or among evolutionary lineages. Option D, rate of DNA or protein sequences evolution is not constant over time or among evolutionary lineages. They are asking the direct question, what are molecular clocks meaning anta kelidare. If you know what are molecular clocks, you can answer this very easily. Molecular clocks are nothing but DNA ಮತ್ತೆ protein sequence evolve ಆಗೋದ್ರ ಬಗ್ಗೆ ಇದು ಹೇಳುತ್ತೆ. Okay, those are that actually called as molecular clock. It represents the rate of DNA or a protein sequence evolution is constant over time and among evolutionary lineages. Option A is the correct answer because that is the definition here. Blue tongue virus causes high mortality among option A fish, option B human, option C sheep, option D poultry. The correct answer for this is option C, that is sheep. Okay, blue tongue virus ಅಂತಂದ್ರೆ as you can see in the diagram here, this is the sheep. tongue ನ ಅಬ್ಸರ್ವ್ ಮಾಡಿ ಅದು ಬ್ಲೂ ಕಲರ್ ಆಗಿದೆ. this is because ಈ ಶೀಪ್ಸ್ ಏನ್ ಮಾಡ್ತವೆ, ಮಿಡ್ಜಸ್ ಅಂತ ಇರ್ತವೆ. ಮಿಡ್ಜಸ್ ಅಂದ್ರೆ ನಥಿಂಗ್ ಬಟ್ ಆ ಟೈಪ್ ಆಫ್ ಮಸ್ಕಿಟೋ ಟೈಪ್ಸ್, ಆತರ ಇನ್ಸೆಕ್ಟ್ಸ್ ನ ಏನ್ ಮಾಡ್ತವೆ, ಗ್ರಾಸಸ್ ಅದು ಫುಡ್ ನಾವು ತಿನ್ಬೇಕಾದ್ರೆ ಅವ್ರ ಜೊತೆಗೆ ಇನ್ಸೆಕ್ಟ್ಸ್ ನ ತಿಂದಿರುತ್ತೆ. ದಟ್ ಆಕ್ಚುಲಿ ಕಾಸಸ್ ಅ ವೈರಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಬ್ಲೂ ಟಂಗ್ ವೈರಸ್. ಸೊ ಟಂಗ್ ಎಲ್ಲ ಏನಾಗ್ಬಿಡುತ್ತೆ, ಬ್ಲೂ ಆಗುತ್ತೆ. So that affects the health of the sheep and it affects other kind of ruminants. Okay, all kind of ruminants and it is more in sheep. Okay, so question ಅಲ್ಲಿ ಯಾವ್ದ್ರಲ್ಲಿ ಅಂತ ಕೇಳಿರೋದ್ರಿಂದ, so ಹಾಗಾಗಿ the correct answer for this is sheep. That is option C is the correct answer. Moving on to the next question. Gondwana land includes the following. Option A North America, option B Europe, option C Antarctica, option D India. Gondwana land includes the following anta kelidre. What is this Gondwana land? We will know. As you can see in the picture that I have displayed. So these are the Gondwana area. Okay. So and this part. Okay. This part Gondwana. This is Gondwana area. And Gondwana, it's a forest of Gondas anta helbodu. What are Gondas? Gondas means other peoples who are living in forest. Okay. Gondwana. It's an ancient supercontinent that incorporated in the present day. Like it includes the South America. So okay, it includes the South America, Africa, India and Madagascar and Australia and Antarctica. ಕ್ವಶನ್ ಅಲ್ಲಿ ಡೈರೆಕ್ಟ್ ಆಗಿ ಕೇಳಿರೋದು ಯಾವ್ದು ಅಂತಂದು ಹೇಳ್ಬಿಟ್ಟು. ಸೊ ಯಾವ್ದೆಲ್ಲ ಇನ್ಕ್ಲೂಡ್ ಆಗಿದೆ, ಇದಕ್ಕೆ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಬಂದ್ಬಿಟ್ಟು 2 ಆನ್ಸರ್ಸ್ ಕರೆಕ್ಟ್ ಆಗುತ್ತೆ. ಅಂಟಾರ್ಟಿಕಾನು ಇದೆ, ಆಪ್ಷನ್ ಸಿ ಇಸ್ ಆಲ್ಸೋ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಈವನ್ ಇಂಡಿಯಾ ಇಸ್ ಆಲ್ಸೋ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ಸ್. So these two are included in the Gondwana land. North America alla, South America answer ಆಗುತ್ತೆ. Europe ಕೂಡ ಅಲ್ಲ. Okay, South America, Africa, Arabia, Madagascar, India, Australia and Antarctica. These are all comes under the Gondwana land. Gondwana, what is Gondwana land? Gondwana land is, it is an ancient supercontinent ಅಂತ ಹೇಳ್ಬೋದು. It's a forest of Gondas. Okay, tribe peoples ಇರ್ತಾರಲ್ಲ, okay, Gondas ಅಂತ ಹೇಳ್ತೀವಿ. To the 43rd question. Which of the following cannot be diagnosed by amniocentesis? So amniocentesis means, what is amniocentesis? Amniocentesis means it is a test or you can say it is a prenatal test. It is done to remove amniotic fluid to test the genetic disorders. Okay, ಇದನ್ನ pregnant woman ಗೆ amniotic fluid ನ ತಗೊಂಡ್ಬಿಟ್ಟು ಟೆಸ್ಟ್ ಮಾಡ್ತಾರೆ ಡ್ಯೂರಿಂಗ್ ಪ್ರೆಗ್ನೆನ್ಸಿ ನಲ್ಲಿ. ಯಾಕಂದ್ರೆ ಟು ಫೈಂಡ್ ಔಟ್ ದ ಫೀಟಲ್ ಅಬ್ನಾರ್ಮಾಲಿಟೀಸ್ ಅಂತ ಹೇಳ್ಬೋದು. ಏನಾದ್ರು ಬರ್ತ್ ಡಿಫೆಕ್ಟ್ಸ್ ಇದ್ರೆ ಅದನ್ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡಕ್ಕೆ, ಅದನ್ನ ಟ್ರೀಟ್ ಮಾಡಕ್ಕೆ ಈ ಟೆಸ್ಟ್ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಫೀಟಲ್ ಅಬ್ನಾರ್ಮಾಲಿಟೀಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಶುಡ್ ರಿಲೇಟೆಡ್ ಟು ಜೆನೆಟಿಕ್ಸ್ಗೆ ರಿಲೇಟ್ ಆಗಿರ್ಬೇಕು. ಸೊ ಅಂತ ಹೇಳೋದಾದ್ರೆ ಡೌನ್ ಸಿಂಡ್ರೋಮು, ಸಿಸ್ಟಿಕ್ ಫೈಬ್ರೋಸಿಸ್ ಅಂಡ್ ದೆನ್ ಸ್ಪೈನಲ್ ಬೈಫಿಡ್. ಓಕೆ, ಸೊ ಈ ತರ ಎಲ್ಲಾ ಟೆಸ್ಟ್ಗಳಿಗೆ ಆಮ್ನಿಯೋಸೆಂಟಸಿಸ್ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ, ಈ ತರ ಡಿಸೀಸಸ್ನ, ಜೆನೆಟಿಕ್ ಡಿಸೀಸಸ್ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡಕ್ಕೆ. ಓಕೆ, ಇದರಲ್ಲಿ ಆಪ್ಷನ್ ಬಂದ್ಬಿಟ್ಟು, ಇದಕ್ಕೆ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಬಂದ್ಬಿಟ್ಟು ವಿಚ್ ಆಫ್ ನಾಟ್ ಅಂತ ಕೇಳಿರೋದ್ರಿಂದ ಸೊ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಇದಕ್ಕೆ ಆಪ್ಷನ್ ಡಿ. ಪೋಲಿಯೋನ ನಾವು ಆಮ್ನಿಯೋಸೆಂಟಸಿಸ್ ಇಂದ ಕಂಡುಹಿಡಿಯಕ್ ಆಗಲ್ಲ, ಆಯ್ತಾ. ಸೊ ಅದರ್ ವಿ ಕ್ಯಾನ್ ಫೈಂಡ್ ಔಟ್. ಓಕೆ, ಬಿಕಾಸ್ ದೀಸ್ ಆರ್ ಆಲ್ ರಿಲೇಟೆಡ್ ಟು ಜೆನೆಟಿಕ್ಸ್. ಮೂವಿಂಗ್ ಆನ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Which of the following plant that existed abundantly during Mesozoic era is considered as a living fossil? So direct question ide, I have to have an idea about this. For option A ginkgo biloba, option B pinus, option C taxus, option D Glossopteris. The correct answer ಬಂದ್ಬಿಟ್ಟು option A is the correct answer, ginkgo biloba. So this is the living fossil. This is the plant which is there during the Mesozoic era. Okay, and this is the living fossil during that time. Moving on to the next question. Plant movements that takes place in response to contact stimuli is called option A thigmotropic movement, option B thermonastic movement, option C chemotrophic movement, option D hydrotrophic movement. So ಇಲ್ಲಿ ಅವ್ರು ಕೇಳ್ತಿರೋದು ಪ್ಲಾಂಟ್ ವಿಚ್ ಮೂವ್ಸ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಇನ್ ರೆಸ್ಪಾನ್ಸ್ ಟು ಕಾಂಟ್ಯಾಕ್ಟ್ ಸ್ಟಿಮ್ಯುಲೈ ಅಂತ ಹೇಳಿದ್ರೆ. ಇದಕ್ಕೆ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಹೇಳಿದ್ರೆ, ಇಫ್ ಐ ಹ್ಯಾವ್ ಟು ಗಿವ್ ದ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಫಾರ್ ದಿಸ್ ಇಸ್ ಟಚ್ ಮಿ ನಾಟ್ ಪ್ಲಾಂಟ್. ಮುಟ್ಟಿದರೆ ಮುನಿ ಅಂತ ಹೇಳ್ತಾರಲ್ವಾ, ಅದು ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್. ಏನ್ ಮಾಡುತ್ತೆ, ಕಾಂಟ್ಯಾಕ್ಟ್ ಇಂದ ಆ ಪ್ಲಾಂಟ್ ಮೂವ್ಮೆಂಟ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳ್ತಿದ್ರೆ. ಅಂದ್ರೆ ಈ ಟಚ್ ಮಿ ನಾಟ್ ಪ್ಲಾಂಟ್ ಟಚ್ ಮಾಡಿದ ತಕ್ಷಣ ಎಲೆಗಳೆಲ್ಲ ಮುದುರುತ್ತೆ. ಓಕೆ, ಅಂತ ಮೂವ್ಮೆಂಟ್ಗೆ ಏನಂತ ಕರೀತಾರೆ ಅಂತ ಹೇಳ್ತಾ ಇರೋದು. ಸೊ ಇದಕ್ಕೆ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಬಂದ್ಬಿಟ್ಟು ಆಪ್ಷನ್ ಎ, ತಿಗ್ಮೋ ಮೀನ್ಸ್ ಟಚ್. ಓಕೆ, ಟ್ರೋಪಿಕ್ ಮೀನ್ಸ್ ಮೂವ್ಮೆಂಟ್ ಅಂತ ಹೇಳ್ಬೋದು. ಓಕೆ, ತಿಗ್ಮೋ ಮೀನ್ಸ್ ಟಚ್. ಸೊ ಟಚ್ ಇಂದ ಮೂವ್ಮೆಂಟ್ ಆಗೋದನ್ನ ತಿಗ್ಮೋಟ್ರೋಪಿಕ್ ಮೂವ್ಮೆಂಟ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅಂಡ್ ಕಮಿಂಗ್ ಟು ದ ಆಪ್ಷನ್ ಬಿ, ಥರ್ಮೋ ಮೀನ್ಸ್ ಹೀಟ್ ಆಗುತ್ತೆ, ಕೀಮೋ ಮೀನ್ಸ್ ಕೆಮಿಕಲ್ ಆಗುತ್ತೆ, ಹೈಡ್ರೋ ಮೀನ್ಸ್ ವಾಟರ್. ಸೊ ಹಾಗಾಗಿ ಇಲ್ಲಿ ಕಾಂಟ್ಯಾಕ್ಟ್ ಸ್ಟಿಮ್ಯುಲೆ, ಕಾಂಟ್ಯಾಕ್ಟ್ ಅಂದ್ರೆ ಏನು, ಟಚ್. So context stimuli ಆಗಿರೋದ್ರಿಂದ option A is the correct answer. Okay guys, I hope you enjoyed this. In the next video, I'm going to solve some other questions. Hoping that this could help you all. Thanks for watching.